Good morning, all of you. Welcome to our session. Today we'll be discussing the most probable questions that may be likely to be asked for the NEET examination this year from chapter number 32. That is second year, first chapter in zoology, human reproduction. That is the chapter from which we are now conducting the session. Human reproduction. Okay, so what will be the thrust area that you have to give more importance to, and what type of questions you can expect are all mentioned here. Now we are going to the questions directly. The first question, odd one from sets. We are given 5 sets P to T, and one of them is not matching with others. That is why it is called odd one, which is not matching with the others. Which is that one? Select that one. Rected testis, of course, that's a part of the male reproductive system. Vasa efferentia from rected testis arises few number of vasa efferentia. They will be all joining to form epididymis. And infundibulum. Infundibulum is not a part of the male reproductive system. Vas deferens. Epididymis continues of the vas deferens, male duct. So, the one which is not matching with the others, which is different from others, is infundibulum. Similarly, prostate gland, Cowper's gland. These are all axillary reproductive glands associated with the male reproductive system. Seminal vesicles, the largest gland, a pair of them. Glands of Tyson. This is not in our NCERT book. Glands of Tyson are present at the tip of the penis. They will be producing a secretion that will form what is called smegma. Okay. Glands of Tyson present at the glans penis. Whereas Bartholin's gland is a gland, the only gland present in the female reproductive tract. All others are on the male reproductive tract. Bartholin's gland is on the female reproductive gland. That is why we take it as the odd one. Spermatocyte, cells that are all developing into the gamete. Spermatid, spermatozoa, Sertoli cells. That is not at all developing into. Instead, it is a part of the testis that nourishes. Okay. Developing spermatids. Spermatogonia cells. So all of these are stages in the development of spermatozoa, whereas Sertoli cells are a structure, type of cell that form the wall of the seminiferous tubule. Vagina, ejaculatory duct. Vagina is a female reproductive tract and ejaculatory duct is a part of the male. Okay. Fimbriae, oral fimbriae, part, the first part of the oviduct. Ampulla, the second part. Cervix. So these are all part of the female reproductive tract, whereas ejaculatory duct is a part formed by the fusion of the last part of vas deferens and the duct from seminal vesicle. Granulosa cells, primary oocyte, oogonia, secondary oocyte, polar body. These are all part of the development of a female gamete ovum, whereas granulosa cells are never developing into an ovum. It is giving nourishment, protection, covering to the developing ovum. So that is the odd one. So, infundibulum, Bartholin's, Sertoli, ejaculatory duct, granulosa. That is D, E. So that is the order. Clear? That's how we came to the answer. So, in each set, what is the odd one which is not matching to the other? We have to select, and what is the set which is containing that set of unmatching structure. That's how we came to the answer. Clear? Spermiation. What is spermiation? Spermiation is the separation of a fully developed spermatozoa from the Sertoli cells. Till that time, it is remaining attached. Okay. So, it is the release of sperm from seminiferous tubules. Till that time, it is attached to the seminiferous tubules. Sertoli cells are giving attachment, providing nourishment. Once this spermatozoa is fully formed, they get detached. That detachment is called spermiation. So one is yes. So you eliminated the other options, 1 and D. Now we came to only 3. Gametogenesis. What is gametogenesis? Simply formation of gametes. No problem. Spermatocytogenesis. Spermatocytogenesis simply means the formation of spermatocytes. Okay, genesis means synthesis. Insemination. Release of sperms in the female genital tract during coitus is called insemination. And spermiogenesis, that's a term which is referred to or which is explaining the conversion of spermatid to sperm. That is a metamorphic change. Spermatid is undergoing a thorough change so that it becomes developed into sperm. That differentiation of spermatid to sperm is called spermiogenesis. It is also called spermateliosis. It is the second stage in the development of sperm. There are 2 steps. First one, development of spermatids, formation of spermatid. Then that spermatid is to go metamorphic change, whereby they become highly elongated, motile sperm. That development is called spermateliosis or spermiogenesis. So the option is box. Flow chart of spermatogenesis is given. You can see, identify the labeled cells during gametogenesis. Spermatogonia cells are formed as a result of repeated multiplication. Large number of spermatogonia cells are formed and they will be developing into primary spermatocyte. That primary spermatocyte is the cell that become or that is ready for meiosis. It undergoes meiosis 1. So here it is first meiosis. So the cell that undergoes first meiosis is primary spermatocyte. And the daughter cells produced after first meiosis is the secondary spermatocyte. Understand that. This is a special condition. Primary is 2n, whereas secondary spermatocyte is haploid. The first cell that is haploid is secondary spermatocyte. In between the primary and secondary, that is during the first meiosis, chromosome number is reduced to half. Diploid is becoming haploid. Secondary spermatocyte undergoes the second meiosis, so that you will be having a total of 4 spermatids. Okay, from one primary spermatocyte, it undergoes first meiosis, so that we will be having 2 equal sized haploid secondary spermatid, and each of them undergoes the second meiosis, and thus you will be having a total of 4 spermatids. Okay, so this is the primary and this is the secondary spermatocyte, and 4 spermatids are produced at the end of the second meiosis. Clear? So the option should be X primary spermatocyte, Y secondary spermatocyte, Z spermatids. Clear? Identify the hormone A and B according to their changing levels during their menstrual cycle. You are given the menstrual cycle. Cycle given that lasts for 28 days, and it is on the 14th day ovulation is occurring. So this is the ovulation date. Okay. Just before ovulation, you can see a hormone is increasing its level. Okay. It is slowly increasing, reaching its maximum, again declining, and again having a second peak by about 22nd to 24th day. This is because completely granulosa sites are damaged and they are completely absorbed. So granulosa cells are producing which hormone? Estrogen. It is the estrogen level that is slowly increasing, reaches the maximum just before ovulation. Okay, just before ovulation. Okay, and now it becomes corpus luteum. After ovulation, it becomes corpus luteum, yellow mass. It starts producing the hormone progesterone. So the B should be progesterone because it is not at all increasing till ovulation. Only after ovulation it starts its peak and reaches the maximum by about the 22nd to 23rd day. Corpus luteum will be remaining active for about 1 week. By that time, implantation should have occurred and the placenta, actually it is the chorionic villi that are all getting implanted and will be starting producing the chorionic gonadotropins. Okay, they will be starting HCG, human chorionic gonadotropins. And this gonadotropin will be sustaining corpus luteum. If no implantation had occurred, if no fertilization, no implantation had occurred, HCG is not produced, and so in absence of that HCG, corpus luteum is not maintained. Now the corpus luteum atrophies, it becomes a white mass, corpus albicans, and completely is absorbed. So the progesterone level decreases after 24th day. And by about the 25th day, when the progesterone level decreases, separation of the endometrium, proliferated endometrium start, and that separated endometrium is flowing out by about the 28th day. It starts separation by about the 25th day. So this hormone, second hormone, which is having the peak by about 20 to 22nd day, will be the progesterone. Clear? So A should be estrogen, B should be progesterone. Clear? This is a diagram which many of you may not have seen. See, when a question which is having a diagram is given, we have to first understand what is that. This is the developing ovum and there's a cavity called antrum. This antrum develops by the tertiary follicle stage. Okay, and antrum becomes filled with a fluid called lakhs folliculi. It is filled with lakhs folliculi. Now the theca will be divided into a theca externa and theca interna. So in between you have a fluid filled cavity. Now the ovum is remaining bathed in that fluid. Okay, and is connected to the wall of the ovary by means of a small part which is called cumulus oophorus. These are all occurring during tertiary follicle stage. In tertiary follicle stage, three important events are taking place. What are they? The theca become divided into a theca externa and theca interna. There will be an antrum, a cavity, which become enlarged and completely occupy almost all part and become filled with a fluid called. That fluid is filled, secreted by the follicle cells. That is called lakhs folliculi. And the third and the last stage is that it undergoes the remaining stages of meiosis 1. It is during this stage that meiosis 1 is complete and the first polar body is eliminated here. And enter into the pro-metaphase and is arrested there at that stage. Clear? So, oocyte adhere to the wall of the graphene follicle. This is the graphene follicle and it is attached by means of this part called cumulus ooferus. Theca externa and theca interna are the two layers. Theca externa, theca interna. And tunica albuginea. That's a connective tissue covering, white connective tissue covering for both testes as well as ovary. Okay. Laker follicle is the fluid that is filling that cavity antrum. Now, hormones labeled as X, Y, and Z. What is that? This is connected with the menstrual cycle. You are having the 4 stages in menstrual cycle. You know that day 1 to 5, that will be the flow. That is called menstrual flow, menstruation. Day 5 to 13, it is the proliferative stage or follicular stage. And here, why the proliferation is occurring? Anterior lobe of pituitary is releasing Z that act on follicle, and follicles are all growing. What is the hormone of the anterior pituitary that will act on follicle cells? It is FSH. That's why the follicles are all developing one by one. The primary follicle, which are all arrested, become developed into secondary, tertiary, and graphene follicle. And it will be completed in 14 days. And now the follicles are all starting their secretion. Follicles are producing estrogen. So, under the influence of estrogen, estrogen is having 2 roles. One, it will be inhibiting the secretion of the FSH because there is sufficient amount of FSH, so you need not secrete more. Second one, it acts on the endometrium so that it undergoes proliferation. That's why I told you the second stage, day 6 to 13, that is the proliferative stage or follicular stage under the influence of the estrogen. Okay, now when FSH inhibits the estrogen inhibits the FSH, now it produces the second hormone, LH. The LH level suddenly increases. All on a sudden it increases, and that is called LH surge, causes graphene follicle to rupture. That is what is called ovulation. Ovulation occurs on the 14th day. And that graphene follicle which is ruptured develop into a yellow mass called corpus luteum. Yellow mass, that is the word meaning. Corpus luteum, yellow body. This is producing the pregnancy hormone, progesterone. It is the angel of pregnancy. It is essential for the maintenance of pregnancy. Throughout the period of pregnancy, progesterone is a must. So, it starts producing progesterone, and the progesterone is responsible for maintenance of the endometrium. So, endometrium will not be separated as long as there is progesterone. Now, fertilization may occur sometimes, and if fertilization occurs, within 22nd to 23rd day, implantation will be occurring. Ovum will be released by 14th day, and if a mature sperm is also there, available, ready for fertilization, fertilization will be occurring. It is only a matter of chance. That's why it is stated that all coitus need not result in pregnancy. Pregnancy occurs only if there is a mature ovum and a sperm simultaneously in the female reproductive tract. Ovum is not always present. It will be normally available after ovulation, that is the 14th day, and will be living for maximum 2 days live. So within that, a fertile sperm should be there. Then fertilization will be taking place, and as a result of that, zygote is formed. Zygote start development, and in 7 days it reaches the blastocyst stage. Blastocyst stage will be, by this time, transported through the fallopian tube and will be coming to the uterus ready for attachment, implantation. So implantation will be taking place normally by the 22nd or 24th day. Okay, now that implanted blastocyst that is having the outermost layer called trophoblast layer start producing small microvilli like structures called chorionic villi. This chorionic villi will start producing the HCG, and that HCG is containing progesterone that sustains corpus luteum. Corpus luteum by its own will be living only for 7 days. If there is HCG, it continues to survive and continue to produce progesterone. And as progesterone is continuously produced, the endometrium is retained there and pregnancy is retained. Otherwise, suppose there is no fertilization, what will happen? There is no implantation, and without implantation, there is no HCG. Without HCG, the progesterone secretion by the corpus luteum will be continuing only maximum up to 7 days, okay, of its formation. That is 22nd to 24th day, depending upon the date of fertilization. After that, corpus luteum is atrophying, becoming corpus albicans. So progesterone level declines, and when the progesterone level declines, the thickened endometrium is not maintained, it becomes separated, and that separated endometrium is going out. That is called menstruation. So now we are having the full endocrine orchestra that is regulating menstrual cycle. Is it clear? So what is X? X is progesterone. Sorry, it is X is estrogen here. And what is Y? Progesterone. And what is Z? FSH. That's why we take the option elephant. Now, what are the tissues that are all developing from the 3 germ layers? Remember, we are all triploblastic. We are having 3 germ layers developing: ectoderm, endoderm, mesoderm. These 3 germ layers are developing for the first time during gastrulation. Gastrula is the stage during which these 3 germ layers are established for the first time. Okay. We are having ectoderm that give rise to, in any animal, ectoderm give rise to the outer layer of the skin plus nervous system. So it is giving rise to epidermis and part of the nervous system. Here there is no brain, nerve cord, etc. But we are having a part. What is that? Pituitary gland. Pituitary is a part of the brain, but clearly understand that this pituitary is having anatomically 2 parts: anterior pituitary, posterior pituitary. Anterior pituitary is also called as adenohypophysis. It is developing from the roof of the buccal cavity, which is called Rathke's pocket. Rathke's pocket or Rathke's pouch. This is adenohypophysis. The second part is neurohypophysis. It is a part of extension of hypothalamus. Hypophysis, the posterior pituitary, is an extension of the hypothalamus. It is a part of the brain. That is why I have written clearly for your information, posterior pituitary gland. Okay, so generally we say that pituitary is a part of the brain, and brain is a part that is developing from, nervous system is developing from ectoderm. So we take it as ectoderm. Clear? Now, endoderm give rise to the liver, alimentary canal, respiratory structures, etc. Here there is no such structures, only liver is there. And all other structures, blood, bones, muscles, kidney, are all developing from mesoderm. So gonads, kidneys, heart are all developing from mesoderm. Clear? So the option goes to apple. False statement from the following. Seminiferous tubules of the testis open to the rectal testis through vasa efferentia. You should understand, each seminiferous tubule is a C-shaped. Tube, only diagrammatic representation. All seminiferous tubules will be joining to form a reticular formation called rectal testis. This is rectal testis. It is a reticulum network. Okay. From this rectal testis start a few small ducts. This is called vas efferents, plural vasa efferentia. All the vasa efferentia join to form a single duct, which is actually the vas deferens. That vas deferens is having two parts. The first part is a highly coiled glandular. It is called epididymis. That epididymis itself is having three parts: a caput epididymis, a corpus epididymis, and a posterior-most cauda epididymis. They are lying on the posterior part of the testis. Okay. So, epididymis is having a caput on the dorsal side, on the posterior side, a body, corpus, and in the posterior, cauda. Now, the cauda epididymis continues as a straight, uncoiled tube called vas deferens, which passes through the inguinal canal, reach up the abdomen. So, that is the course. Okay. Now, the question, testis open to the rectal testis through vasa efferentia. No, it is not through the vasa efferentia. See, what actually is correct? Through the vasa efferentia, it is open to the epididymis. Rectal testis, this is the rectal testis. That rectal testis, this is the rectal testis. Through the vasa efferentia, open to the epididymis. That is the correct one. Here it is given, testis open to the rectal testis through vasa efferentia. No, it is wrong. Vasa efferentia leave the testis and open into epididymis. Okay, so that is correct. Epididymis leads to the ejaculatory duct that ascends to the abdomen. No, epididymis descends down, forms the vas deferens. See, epididymis descends down and ascends as the vas deferens. Okay, as the vas deferens, it reaches the abdominal cavity, is joining with the duct from the seminal vesicle and is forming the ejaculatory duct. Epididymis leads to ejaculatory duct. That is the wrong one. Vas deferens receives a duct from seminal vesicle, correct, and opens to the urethra as the ejaculatory duct. That also is a correct one. So, 2 are wrong, 1 and 3. So, the option is doll. Choose the correct combination of labeling the hormonal control of female reproductive system. See, whether in male or female, the hormones are same and they will be having the same action, but the target cells are slightly different. For example, in hypothalamus, which is the master of the master gland, we are having releasing hormones. Only in presence that releasing hormone, the anterior pituitary hormones are coming out. Gonadotrophic hormones are produced by the anterior pituitary. They will be coming out. They will be releasing. Only in presence of GnRH. Okay. So we are having hypothalamus producing gonadotrophic releasing hormone. In presence of releasing hormone, it produces both FSH and LH. Okay. We are having FSH and LH. Now, FSH will be acting on, this is ovary. They will be acting on follicle cells. So, follicle cells are developing primary, secondary, tertiary, then Graafian follicles or ovum is produced. And during the formation of Graafian follicles, follicle cells are producing estrogen, and that estrogen will be acting on uterus. Okay, they are all acting on uterus. And estrogen will be suppressing hypothalamus. We have studied it, and it starts producing the LH, and that LH will be responsible for the release of the ovum. LH surge is responsible for the release, and after that, the corpus luteum is formed. Corpus luteum starts producing progesterone. Okay, so what is A, B, C, and D? A is GnRH. B can be either FSH or LH. Okay, both are produced by the anterior pituitary. And C is always estrogen, and that is acting on D. D is uterus. That is why endometrium of the uterus is undergoing proliferation. Clear? Now, arrange the role of hormones in the menstrual cycle in the correct sequence. We are having for 5. You have to arrange them in the order as they are all occurring. So, you please read it. Corpus luteum degenerates. That is the last part. Of course, we know that it should be the last part. So, it is maybe the 5th one. LH and FSH attain a peak in the middle of the cycle. That is not the first stage. FSH stimulates the follicle development. Okay. LH surge induces the rupture of Graafian follicle. Corpus luteum secrete large amount of progesterone. See, what should be the correct one as they occur? FSH should stimulate the follicular development. Okay, so FSH attain, that should be the correct answer. First, it should be the FSH developing, stimulate the follicular development. Then only FSH and LH attain a very peak in the middle, and LH surge induces the rupture of. Okay, that will be. So the correct one should be R. LH stimulates the follicular development, then LH and FSH attain a peak, then LH will be causing the rupture, and that ruptured one develop into the corpus luteum. That corpus luteum will be secreting large amount of progesterone, and corpus luteum degenerates and causes the disintegration of endometrium after a week. Okay, so after a week of ovulation, that will be occurring at the last stage. So this is the order in which they will be occurring. So R, then Q, S, T, P. You can see it. Both FSH and LH are produced by the anterior pituitary. First, FSH is produced and it reaches a maximum. Why? Because FSH is responsible for the development of follicle cells. That is why follicles are developing into secondary, tertiary, and Graafian follicle. Now, as they are all developing, ovum is developing inside them, and the surrounding follicle cells are causing the release of their hormone estrogen. Estrogen and LH attain a peak by the middle, and it is the LH surge that causes the release of the Graafian follicle, bursting of the Graafian follicle and release of the ovum. So that is the correct option. Consider the statement A to D. Primary oocytes are temporarily arrested cells in the prophase 1 of meiosis. During female gametogenesis, there are two arrests. The first arrest is a longer one. Remember, the process of multiplication takes place even before a girl child is born. All oogonia cells are formed before a girl child is formed. And after her birth, no more oogonia cells are formed or added. Now, they will be developing into, this oogonia cells will be developing into primary oocyte. This primary oocyte undergoes the first meiosis. It starts its first meiosis, completes its first prophase, then it is arrested. That is the first stage. This will be taking place in the early stages. Okay. Now, after the arrest, the remaining stages will be taking place only when she reaches her puberty stage. The remaining stages, main stages of development take place only after she has attained puberty. So, for 10 years, 12 years, these follicle cells are remaining arrested in the ovary. Okay, so primary oocytes are temporarily arrested cells in prophase 1 of, that is the first longest arrest, correct. Granulosa cells, that is follicle cells, and theca are present in primary. Now, I told you earlier that theca is divided into theca externa and theca interna, and theca is actually formed during the secondary follicle stage. During the primary follicle stage, each ovum will be surrounded by a single layer of follicle cells. That is why it is called primary follicle. Only one layer of follicle cells. Now it is primary follicle stage. It gets arrested. So granulosa cells and theca, there is no theca. Theca develops only in the secondary follicle stage. In the further development, only when she has attained puberty, this stage will be undergoing development further to secondary. Then only a theca is formed. And theca is dividing into a theca externa and theca interna in the tertiary follicle stage. So it is wrong. The first meiotic division complete in the secondary follicle and result in secondary oocyte and first polar body. First meiotic division complete in the secondary follicle. Is it in the secondary follicle? No, in the tertiary follicle. It is the tertiary follicle that the primary oocyte is dividing, okay, into a secondary oocyte and first polar body that occur only during tertiary follicle stage, not in secondary. Okay, and tertiary follicle become mature graphene, correct. That is the last stage from primary, secondary, from secondary to tertiary, and tertiary to graphene follicle, fourth stage. Okay, so that is correct. So 1 and 4 are correct. A and D are correct, others are wrong. Clear? In mammals, the penetration of sperm into the ovum is facilitated by an enzyme called, see, we are having on the tip of the nucleus a massive structure called acrosome. This acrosome is a cap-like structure, okay, formed from Golgi complex, which are all concentrated packets of digestive enzymes. When I say digestive enzyme, not our digestive enzymes that we studied in digestion. They are responsible for digesting, breaking down the cell layers, protective coverings around the ovum. That will be helping in penetration of the sperm into the ovum. These enzymes are called acrosines or sperm lysines. Okay, what are these enzymes? They are all corona penetrating enzyme, which is helping in penetrating through the corona radiata layer. Then hyaluronidase. Remember that ovum is having a layer of corona radiata cells around them. Okay, as a protective layer, we are having corona radiata. That corona radiata are all connected together by a cementing substance, namely hyaluronic acid. It is that cementing substance that keep the corona radiata attached to each other. That cementing substance is dissolved, digested, so that one by one the corona cells are removed. The ovum become naked one by one. That enzyme is called hyaluronidase. Third one is acrosins. The third one, which will be acting on the zona pellucida, zona lysine, or acrosin. Will be dissolving the gelatinous layer around the ovum, zona pellucida. So these are all the three enzymes generally called sperm lysins. Okay. Anti-fertilizing is a attractant. By the attraction of or by the specific activity of anti-fertilizing produced by the sperm surface and fertilizing produced from the unfertilized egg surface, the attachment of the sperm and ovum will be occurring. That's why a sperm will be normally attracted by the ovum of the specific species because of the specificity between fertilizing and anti-fertilizing. Luciferin luciferase, that is connected with production of light, emitting light. Hyaluronic acid is the material, substance that cement the granulosa cells around the ovum, and it is digested by the hyaluronidase. Thymase. Thymase is present in some fungi like the baker's yeast, Saccharomyces. Okay, so sperm surface is producing anti-fertilizing. Hyaluronidase digests the cementing substance between the follicle cells, corona radiata, that is hyaluronic acid. So the answer is hyaluronic acid. Meiotic division of the secondary oocyte. Now told you, first meiosis takes place during the primary oocyte, and after first meiosis, that is taking place in the tertiary follicle stage, you will be having one secondary oocyte and one polar body. Now it is getting arrested there. It passes through the prophase 2 only and is getting arrested there. So at the time of ovulation, the ovum has not completed its meiosis. It is arrested at the prometaphase 2. The remaining stages of meiosis will be taking place only when sperm penetration occurs, because the sperm is providing certain chemicals that will activate, that will break that arrest and activate the ovum to undergo the remaining process of meiosis. So only at the time of fusion of a sperm with an ovum, the second meiosis is complete. It is not completing meiosis 2 at the time of ovulation. It has completed only prophase 1, it has completed the first division and has completed the prophase 2 only. Which of the following hormone level will cause release of ovum? We know that. See, in another form I have given you, hypothalamus producing GnRH, it is activating the anterior pituitary to release the FSH. FSH will be activating oogenesis because it acts on the follicle cells. So follicles are developing and follicles are producing estrogen. That estrogen is having two actions. One, it is suppressing the anterior pituitary so that FSH production is stopped and LH production is activated. That LH will be causing ovulation, and after ovulation, corpus luteum is formed. Corpus luteum will be producing progesterone. Okay. The estrogen act on secondary sexual organs and also act on uterus, endometrium, so that endometrium of uterus undergoes proliferation. So, now, which of the following hormone levels will cause release of ovum? High concentration of LH, that is called LH surge. So, understand, under what condition? There is no such answer. There is no such answer. LH surge only is that low concentration. You cannot take it. It is not a mistake actually. You have to think, when will the LH be released? LH is released only by about the 40th day. Suddenly it is releasing and its concentration is increasing all on a sudden. When FSH is sufficient, that follicle cell stimulating hormone is producing the Graafian follicles. Graafian follicles are having follicle cells. Follicle cells are producing estrogen in abundance. So, when there is sufficient amount of estrogen, the anterior pituitary is producing LH. That LH is causing ovulation. So, high concentration of estrogen. It is not directly involved, but that is the situation. High concentration of estrogen causes anterior pituitary to release the LH, and that LH is responsible for the LH surge. That actually is the reason for immediate cause for rupture of the Graafian follicle and release of the ovum. So the correct answer is 4. Don't jump into conclusion. Okay, this is low concentration. Actually, it should be high. So when the high concentration of LH is occurring, when estrogen level is maximum. High concentration of estrogen. Now, match the following columns. Placenta. What is placenta? Placenta is a temporary endocrine gland. Androgens, no. HCG, human chorionic gonadotropin, correct. Placenta is producing. So, 1 is 2. Immediately we came to the answer because there is only one option where A is 2. So, we saved a lot of time. Zona pellucida is a non-cellular gelatinous layer produced by the Graafian follicular cells around the ovum. Bulbourethral gland. Bulbourethral gland are also called Cowper's gland, present in the male reproductive tract, a pair of small structures which are all producing mucus. It is mainly responsible for the lubrication of the penis. And Leydig cells. Leydig cells are interstitial cells in the testis. They are present in the matrix. They are all responsible for the production of testosterone, androgens. So the option is 3. Which of the following events is not associated with the ovulation? LH surge, that is the real cause of ovulation. Decrease in estradiol. See, estradiol is a type of estrogens. Remember, these are all steroid hormones, and steroid hormones are of different types. All of them are having same action. Their functional group is only slightly different. Estrol, OH group is there. That's all. Slight differences. Cortisone, cortisol. Steroid hormones are all like that. Okay. So, decrease in estradiol. See, what is estradiol? I have given you. There are 3 types of estrogens in our body. They are called E1, E2, E3. E1 is called estrone. Okay. It is produced only after menopause. E2 is the most abundant during the reproductive years. It is the strongest. It is almost 80 times more stronger than the estrone. Normally, during reproductive years, that is a girl belonging to or between menarche to menopause. And third one is estriol, that is produced when a mother is pregnant by the placenta. Produced by the placenta. Plentiful, and it is very weak. Okay. So, decrease in estradiol, decrease in estrogen. Okay, you take it as estrogen. There is an increase. Remember, in the previous chart, we have studied that in one of the graphs. Yes. See, estrogen level is increasing and is reaching a peak by about the middle of the menstrual cycle. So it is on the 40th day. The estrogen is increasing. So it is not decrease. Clear? So decrease in estrogen is not correct. Estradiol is not correct. It's increase. Full development of Graafian follicle. Graafian follicle, once it is fully formed, ovulation is occurring and release of secondary oocyte. Secondary oocyte is released. I told you the ovum has not completed the meiosis 2. It is only completed the first meiosis in the stage of secondary oocyte. That is released. So all others are connected with the ovulation, whereas decrease in estradiol is not an important one. It is not at all occurring. It is actually an increase. In human female, meiosis 2 is not completed. I told you second and final meiosis is completed only when sperm penetration occur. Okay. So birth, no. Puberty, no. At the time of fertilization only. When the sperm penetration, because the sperm penetration will stimulate the ovum to undergo further developments. Okay. It activates an ovum. Suppose there is no fertilization, the ovum will be remaining in that dormancy, sleeping condition and will be perishing. It is not undergoing the second meiosis. It is not completing if it is not fertilized. It is the fertilization that activates an ovum. Which of the following layers is an antral follicle? Is acellular, which is not made up of cells. Zona pellucida. See, the zona pellucida is gelatinous. It is made up of mucopolysaccharide. It is non-gelatinous. Sorry, it is non-cellular. That's why this is the answer. Granulosa cells, the cells that are all around it. Theca interna and theca externa. Stroma is a connective tissue matrix. Stroma is a connective tissue matrix which is having cells in it. So the only acellular antral follicle, following the layers in antral follicle. So antral follicle are developing only in the tertiary follicle stage. So here it is not a part, stroma is not a part of it. Theca externa, theca interna are made up of cells. Granulosa cells are cells. Zona pellucida. So zona pellucida will be formed in a Graafian follicle only after that in the Graafian follicle. Which of the following depicts the correct pathway of transport of sperms? You are having C-shaped seminiferous tubules which are all joining to form a rectate testis. From rectate testis arises the epididymis. From epididymis you will be having the highly coiled and glandular epididymis. Okay, epididymis. This epididymis is continuing as the non-coiled structure called vas deferens. So that is the order in which sperms are all passing. Match the following. What is mons pubis? A thick layer of fatty layer. It's a part of the external genitalia of a female. Female external genitalia. You cannot immediately come to the answer because there are three options. You can eliminate only one. Antrum. Where is the antrum? Antrum is produced during development from tertiary follicle onwards. Here there is no tertiary, so it is Graafian follicle. Trophectoderm. Trophectoderm is the outermost layer, okay, of the blastocyst. that will be getting attached. So it is embryo formation. Actually, it is not from trophectoderm that embryo is formed. Trophectoderm give rise to the structures that form the placenta and extra-embryonal membrane. It is concerned with nourishment, and inner cell mass is containing stem cells that undergo proliferation and give rise to the germ layers, and embryo develop from inner cell mass. Okay, and trophectoderm is providing nourishment for the development of sperm, for the development of the embryo. That's all. What is Nibbon kern? Nibbon kern is a mitochondrial cloud that is present in the middle piece of the sperm. It is very often asked. That word is not in our NCERT book. Nibbon kern is a mitochondrial cloud present in the middle piece of the sperm, which is spirally coiled around the axial filament. That Nibbon kern is, that is the only match sperm. So we have to take it. Okay, so the option is 4. Several hormones like HCG, HPL, estrogen, progesterone are produced by H. It is the human chorionic. See, chorion is the outermost layer, which is a part of the placenta. So it is placenta that is producing all these things. Placenta is a temporary structure which is also endocrine in function. It produces a number of hormones, and HCG and HPL are very, very important one. What is capacitation? That's a very important process taking place within the genital tract. Once the sperm is released, that is insemination occur during coitus, the semen is released to the female genital tract. That sperm is not ready for fertilization immediately. It has to undergo a lot of physiological changes. The overall changes that sperm is undergoing, so that it attains the ability to undergo fertilization, is called capacitation. This is a unique feature in mammals. Only then the sperm will be getting the ability to fertilize ovum. This takes place in female reproductive tract because only after insemination the sperms are released to the female genital tract. Capacitation, that's a long process. It will be taking 5 to 6 hours. Then only the sperms will be getting the ability to fertilize it. Clear? A temporary endocrine gland, that's what I have told you, is the corpus luteum. It will be active normally if there is no fertilization for about 1 week, and if fertilization had occurred till baby is released out. So that's corpus luteum. Pineal gland is a permanent gland that produces that acts as the biological clock. Corpus caardiacum allatum system is actually a part of the endocrine system in insects. In insects, we are having neurosecretory cells in brain that produces secretions that are stored in corpus caardiaca, which is responsible for the development of the body, increase in growth, and heart control thereafter. Whereas corpus allatum and prothoracic gland present in the first thoracic segment of the larva, that is producing ecdysone, prothoracic hormone. Prothoracic gland produces ecdysone. It's a steroid hormone that produces, or that is essential for molting, whereas corpus allatum is responsible for formation of a hormone called juvenile hormone JH, which is responsible for maintaining the larval stage. So when ecdysone is abundant, molting occur. When JH hormone is abundant, the larva will be remaining in that larva stage itself. That's how the larva is molting into next stage when JH hormone is low and ecdysone hormone is high in concentration. That is the system in insects. Okay. Why the corpus luteum is an endocrine? Because it is responsible for the production of progesterone. Clear? Amnion of mammalian embryo. Remember, we are having 4 membranes around the fetus. In mammals, we call it as extrafetal membrane or simply fetal membranes. Whereas in other amniones like reptiles and birds, we simply call it as extra-embryonal membranes. These membranes are all produced outside the developing embryo. That's why it's called extra-embryonal. They are all present in all amniotes. Okay, we are having 4 membranes. What are they? They are all the outermost chorion, innermost amnion. Both of them are developing from ectoderm and outer layer of mesoderm. Parietal layer of mesoderm. Somatopleuric extra-embryonic mesoderm on the outside, and we are having trophoblast layer, that is ectoderm, on the inside. Chorion is having the same layer but in an inverse order. So there the trophoblast is outside and somatopleuric. What is somatopleuric? Somatopleure and splanchnoplure are the 2 layers of the mesoderm. Okay, see, mesoderm is splitting. Outer is the somatic and inner is splanchnic. So somatopleuric extra-embryonic mesoderm outside. Clear? Now, the other 2 membranes. So I told you about the chorion, the outermost, and the innermost amnion. You are having allantois and the yolk sac. They are all formed from endoderm and inner mesoderm. Endoderm and inner mesoderm. What is that inner mesoderm? Outer one is called somatopleure. Now it is splanchnoplure. Splanchnopluric extra-embryonic mesoderm. In yolk sac, endoderm is inside and splanchnopluric mesoderm is outside. The reverse is for allantois. In allantois, splanchnopluric extra-embryonic mesoderm is on the inside and the endoderm is on outside. So simply understand, it is from ectoderm and outer mesoderm that we are having the chorion and allantois. The order is different. It is just reverse. Similarly, it is from inner mesoderm and endoderm that we are having the chorion, allantois and yolk sac. Here again, the 2 layers are in opposite directions. If in one it is like this, the other one it is in a reverse order. Clear? So, if you want, I will explain once again. Chorion and innermost amnion. Both of them are having mesoderm, outer mesoderm, that is called somatopleure. Somatopleure outside and chorion, and inside you are having the ectoderm. Okay, ectoderm is trophoblast. In chorion, it is just the reverse. Ectoderm, that is the trophoblast, is on the outside, and somatopleure is inside. Clear? Now, yolk sac. In yolk sac, it is not somatopure, it is planktopure. Outside and endoderm inside. In allantois, it is just the reverse. You are having the endoderm outside and planktopure. Planktopure is the inner mesoderm. Mesoderm is splitting into. We know that we are all having mesoderm which is splitting. So planktopure which is on the inside. Now you got the layers. Okay. This is the condition in all amniotes. Okay, in all amniotes. Hormone secreted by the placenta to maintain pregnancy. So during pregnancy, what are the hormones that are produced? HCG, HPL, that is responsible for the development of the mammary glands. Progestogens, that will be supplementing estrogens. HCG, HPL, estrogens. Relaxin. Relaxin is produced by the ovary, responsible for the increase in the birth canal, dilation of the or softening of the pubic symphysis, so that birth canal is increased. So that is by the ovary. HCG, HPL, progestogen and prolactin. See, prolactin is produced by the anterior pituitary, responsible for the development of mammary glands. That is the beginning of puberty. HCG, progestogen, estrogen, glucocorticoids. Okay, so the correct answer will be 1. Difference between spermiogenesis. What is spermiogenesis? In the first question we have discussed the spermatids are undergoing a transformational metamorphic change, that is called spermiogenesis or spermatoeliosis. So that spermatids are converted into highly specialized sperms. Okay. Whereas spermiation is the separation. Fully developed sperms are all separating from the Sertoli cells because they have completed the development. Till this time, they are all remaining attached by their head to the Sertoli cells. Sertoli cells are nourishing cells. And from the Sertoli cells, they are all taking nourishment and they are all growing. And after completing their spermatoeliosis, they become separated. That separation is called spermiation. So, in spermiogenesis, spermatozoa are formed. Correct. Well, in spermiation, the spermatozoa are released from the Sertoli cells to the cavity of the seminiferous tubules. That is the correct one. Match the following. We are having 3 stages. Proliferative stage. What is proliferative stage? During which the endometrium is proliferating. And it is because of the development of estrogen. So estrogen is continuously produced. Why? Because it is Graafian follicles growing. So it is also called follicular stage. What is secretory phase? Graafian follicle is secreting progesterone. So it is during this stage that progesterone is produced. And it is also the luteal phase because corpus luteum is formed. It is secreting. So B is 3. And menstruation, that is the breaking down of extra thick proliferated endometrium. Only endometrium is undergoing such changes and is broken down during menstrual cycles. Correct sequence for transport of sperms, we have studied it. In male reproductive system, we have discussed this. Still, I will tell you what is that. They are produced in the seminiferous tubules of the testis. They will be reaching the rectate testis. Then, you are having vasa efferentia, large number of small tubes, 10 to 12 such small tubes are there in each testis by which they will be collected, and these vasa efferentia are joining to form the glandula, highly called epididymis. Then it becomes vas deferens, single. There are two vasa deferentia from one testis each, only one, and ejaculatory duct, urethras. Remember, in males, the urethra is a common passage for both sperm and the urine. Finally, through the meatus, urethral meatus, they are all opening out. They are all, both of them are released through the meatus. So that is the correct option. Extrusion, that is release. Release of second polar body from egg. See, it is during the second meiosis. That second meiosis completed only at the time of fertilization. Secondary oocyte is undergoing incomplete division in both first and second meiosis. Cytoplasmic division is highly unequal, so that you will be having one large cell and a very small polar body. In the second division, the polar body is called second polar body. So in secondary or in second meiosis, secondary oocytes, secondary oocytes, it is not oocyte, oocyte, is undergoing the meiosis II, producing one large haploid ovum and one polar body. That occur only after the entry of sperm, but just before fertilization. Sperm contact is activating. Colostrum, the yellowish fluid secreted by the mother during the initial days of lactation, that is very thick, yellow colored, is very essential to the important immunity to the newborn, because through that mother is producing antibodies and giving passively. It is a typical example for natural passive immunity. IgA, immunoglobulin A. It's a dimer. It is called secretory antibodies, and secretory antibodies are abundant and it contains all the essentials in a readily absorbable form. That is the ideal food for baby. So the baby should be given this milk. And remember, immunoglobulin A is secreted through saliva, tear, etc. and will be also sent out of the body through our feces. That is why it is called copro antibody. Copro antibody, IgA, it's a dimer having two monomers. So a total of 8 chains are there, 4 heavy and 4 light chains. Okay, it is a dimer called also a secretory antibody, copro antibody. Okay, and with that we come to the conclusion of the 30 questions from this chapter. I hope it is thorough to you. Okay, now let me conclude this chapter and say goodbye to you. We will be meeting with the next set of questions on another day. So thank you all. Thank you very much.